Hi. I'm leaving and I'm chairing the session on the short talks, so I would invite Claire Rioualen to come here to give the first talk on ELIXIR. And I will just give you a mic so that we can get started. As before, I will stand up at about 8 minutes, so that you, yeah, turn on. It's, yeah, it should work.
Okay. Can you hear me? Yeah. Okay. So I'm Claire. I work at the French Institute of Bioinformatics, which is also the French node of the ELIXIR infrastructure. And today I'm going to talk about bioconductor software integration into the ELIXIR Research Software Ecosystem through metadata standardization. Okay, so basically I'm going to talk about what the research software ecosystem is and why integrating bioconductor software package metadata in the ecosystem can benefit users and developers. By answering questions very simple, such as which package can perform the functions that I need, or how can I help users find and use my package. So, so as many of you know by now, there's a collection of more than 2,400 software packages in Bioconductor. They are annotated with BioCVUs, which is a controlled vocabulary. It includes almost 500 terms and 175 that are software specific. But they are not used very consistently. The terms are used between 0 and 800 times. And the packages are annotated with between 0 and 45 terms. Um, and yeah, no. But I missed one. Okay, so software package annotations include 238 terms, and they are not necessarily in BioCVUs. There's 52 terms that are not in BioCVUs. There are 25 terms in the software annotations that are not software specific, and there are BioCVUs terms that are not used at all. So we decided to use the ontology for the annotation of packages. The ontology is it defines concepts in life science data management and analysis, and it has four main classes, which are topics, operations, data, and formats. And and I don't understand why it's not. Oh yeah, okay. And those classes have semantic relationship. Operations have input and output of class data. Data have formats associated, and operations and data have topics. Um, yeah. So first, we try to map the BioCVU's vocabulary and the package annotations to EDAM. About 72% of the terms could be could be matched. About 15% would be candidates for new concepts in EDAM, and about 12% were out of the scope of EDAM. And most of the terms that matched were matched to operations and topics. So that's work we did during the 2024 ELIXIR Biohackathon. And yeah. And so EDAM is used to annotate tools in the BioDataTools registry. BioDataTools contains more than 30,000 entries for tools that are annotated with EDAM concepts, not only, also other ontologies are annotated. So the tools in the registry can be annotated with lots of EDAM concepts and also publications, repositories, quality metrics, that sort of things. And so as an example here, I put the page for the XCMS package, which I will use over the presentation as a use case. And it has a couple of topics, EDAM topics, which are metabolomics and data visualization, and a bunch of operations and input and outputs. Yeah. So, okay, I'm gonna manage. So BioTools is a gateway to the research software ecosystem, which imports and standardizes metadata from different sources, just like Bioconda, Galaxy, Workflow Hub, the biocontainers, and so on. And the idea is to uniformize those metadata to be able to integrate all together. And so we want to incorporate, incorporate bioconductor package metadata into this ecosystem. So second thing we did was to synchronize the metadata with the ecosystem, and about 68% of the packages were already in the ecosystem, so their metadata were updated. And 32 packages were not there yet, so the entries were created and the metadata were imported. And so we did that a couple of years ago, so it was version, I think it was released 3.20. So yeah, basically we added 700 new packages in the ecosystem. We also did that at the biohackathon in 2024. Okay. These annotations, they can be represented as graphs. So with the example of XCMS, for example, we can annotate topic metabolomics. It has an operation which is chromatogram visualization, which has mass spectrometry data as an input and Plaut as an output. Just a simple example. Now, if we integrate, I really hate this thing. Okay, well, I skipped one, but it's okay. So we integrate the bioconductor metadata, some of it anyway, a couple of more information, and we can query those graphs. So we can, for example, here ask which tools perform chromatogram visualization, and we would have two answers. Which topics are associated with XCMS, that would be metabolomics. And if we operate more information, yes, that's the next slide. Okay. Okay, so we can add a lot of information from the ecosystem. It's just a tiny sample, but then you can start querying the graph with more complex questions, like what are the dependencies of the XCMS bioconda container and what do they do? Well, that's the spectra container, and it's linked to the tool which performs spectral analysis, and so on and so forth. Yep. So yeah, so in order to have all of this information in this giant graph, there's a lot of curation effort that is involved. It's very time consuming. Sometimes there are inconsistencies. Some tools can be annotated differently in different places. So, so at some point, obviously, we want to try and use LLMs to annotate, to automate the annotation of packages by using, for example, package vignettes or descriptions. But it did not work so well because there are errors and hallucinations. For example, here, when try to get the list of functions that XCMS can produce, perform, there was chromatogram processing, which is, it's not a concept in EDAM, so it shouldn't be here. And also there was an identifier that is not the right identifier. So basically, an hallucination. So to avoid that, we have started to work with a model context protocol approach, which basically allows to have LLMs interact with external tools or resources in a like dynamic way. And so in order to compare approaches. Yeah, okay. So basically, we have developed EDAM MCP, which is an MCP server that can interact dynamically with the EDAM ontology. And we have compared the performance of the EDAM MCP against the simple approach, just ask an LLM. And so we have generated a set of question and answers to use as a ground truth. For example, based on XCMS metadata from BioTools, what topics does it cover? And that would be metabolomics. Now, when we tested the approach with ZMMCP, we had the metabolomics as an answer 5 times out of 5. And with the simple LM approach, it was just twice. And the other answers were either unrelated to metabolomics or it was a concept that did not exist. So yeah, overall, we have better performance using LMMCP than just using LLMs alone. But we have yet to extend the benchmark to investigate that more. So it's also some work that we did. It was mostly developed at the 2025 ISMB CoFest and the 2025 ELIXIR Biohackathon. And so we made a cool poster and a preprint also, which you can see. I put the QR code over there. I realize it's way too small. But anyways, there's a preprint where we describe all of that work. And now, just to conclude, using EDAM for metadata annotation can lead to better consistency of the annotations. Their integration into the research software ecosystem can greatly facilitate curation efforts. The ecosystem, by centralizing metadata, also allows to build and query knowledge graphs. And but overall, it can improve the findability and interoperability of bioconductor tools. There are a few about bioconductor annotation specifically. How could we incorporate semantic relationships into metadata? How to deal with R objects? We've been dealing with input and outputs as file formats mostly. So R objects are a bit different. And which granularity should be used for the annotations? Some packages, for example, they perform a lot of functions. Some are very. Do we want to annotate all of it? Well, in the research software ecosystem, right now we are serializing the metadata of all of the sources that I cited earlier to RDF files, and then we are setting up Sparkle endpoints where anybody can go and query all of this data. Coming soon. The last slide.
Yeah. Let's give Claire a hand. Thanks for managing it with all these hiccups. So in the interest of time, we only have time for one quick question. Okay.
Thanks for the talk. Could you expand a bit on how this Sparkle endpoint would be useful and what kind of information you could fetch from it?
Well, basically, imagine the graph that I showed, but like very, 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 very. I mean, like, basically the Sparkle endpoint, it allows to access a kind of RDF store where you have all of these triplets of information, and we have thousands and thousands of those triplets. The nodes and edges that we had in our graph. So it's like a giant graph that you can query with Sparkle queries precisely. And so yeah, you can ask anything you want to that Sparkle endpoint. Like you will have access to all of the metadata of the researchers ecosystem. Data from Conda, from Biocontainer, from WorkflowHub.
Okay, so let's give Claire a hand again.
I just wanted to say also that I think it's cool that we are working on this, and it's like a bridge between Bioconductor and ELIXIR, which are two great communities, and I hope that there's more.
Okay. Oops. Yeah, normally it works. So Mohammad will introduce an R package to monitor GPUs, and I'm happy to give you the floor.
Okay, thanks. I'm Amin from University of Padua, and what I about to present is an R package that we developed as he told that to monitor NVIDIA GPUs during the R workflows. Just to briefly explain what is the difference between CPU and they have a small number of cores that they're usually very powerful and they are very good at running sequential computation containing if, this, then, that, and they are very good for running operating systems or stuff like this. But on the other hand, GPUs, they have billions of cells that they are not that much powerful, but they are very good at running same operation on a huge amount of data that can be exemplified in matrix operations that they have lots of small operations like summation, multiplication, running through billions of operation at the same time. These are actually the foundation of the PCA, KNN graphs, UMAP clustering, other stuff that they are parts of, for example, of single cell analysis. We use R for all of the analyses that I will show during this presentation, but the backend for all of the computations based on CPU and GPU are in Python. And in Python side, in CPU side, we use ScanPy, that is a library in Python. And for the GPU computation, we use rapid single cell by NVIDIA, that is exact identical version of ScanPy, but utilizing GPU for all the computations. The workflow that we use is actually one of the general, I would say, workflows that we took from the NVIDIA rapid single cell that contains 10 steps. That if you look at this workflow, this not necessarily all of the steps should be same in all the workflows, and there are hyperparameters. For example, have different implementations. KNN, for example, has could have number of neighbors that it considers. So there are hyperparameters over these steps. To run this workflow on CPU, the runtime was like 1 hour and 20 minutes. That in GPU side, it takes like 2 minutes and 30 seconds to run it. And a question may raise that then the running time on the scale of hours, for example, 1 hour, 10 hours, 100 hours, there is no reasonable, I would say, excuse to invest a lot of fortune to have HPCs or servers contain GPUs. But this is not the whole story. If one wants to study different configurations of the workflow that I showed before, and consider that there are just 2 options for steps in the workflow, excluding the loading, because loading is always the same, then in a combinatorial explosion will up with 20,000 pipelines. That if we want to run all of this to just to have the upper bound of the computation, because one will study all of this but to have just the upper bound of the computation, if you want to run all of these workflows on CPU, feasible somehow. So running using GPU is not about developed actually is use NVML, that is a library by NVIDIA, is NVIDIA Mac, that's written in C, or it to R using function calls to also different features as CSV or plotting directly. It is actually very easy. It's like that you start a sampler. Stop the monitor, parse the sampler, and then outside the plot. So it's very easy to use. For case study, we use data come from the 10x Genomics E18 mouse brains contains 1.3 billion cells that they have like 30,000 genes, and we select 1 million, first 1 million cells of this data based on what rapid single cell tutorials done. And we feed the same data to CPU and GPU version of the workflow. We ran the actually CPU workflow on the CPU node of the HPC server that we have in our lab. To be sure that there is no GPU that is utilized during the running code, to be sure that is 100% run on CPU. And the GPU node, we have one of the best actually currently NVIDIA GPUs for HPC servers is NVIDIA H200 that has 140 gigabytes of RAM. If you look at the CPU version and GPU version regarding the CPU, there is a bias introduced here because they are not identical, the CPU in both two nodes. But if it's also a bias is introduced is in the favor of the CPU node because the number of cores is so much more in the CPU node. We designed 3 experiments to better understand what we have take out of this monitoring of the GPU during the running of the workflow. The first experiment is to head-to-head comparison between the CPU workflow and GPU workflow stepwisely. If you look at just at the big picture, we can see that when we utilize GPU, we have 32 times a speed up from like 78 minutes to 2.5 minutes in GPU. But if we look at the steps, we can see that in some computational intense steps like UMAP and clustering that ends up like 1,600 seconds in CPU, they enjoy the most acceleration on the GPU version of the workflow that they ran, both of them below 20 seconds. And it shows that the implementation works because we expect this acceleration be more in the side of these computational intense steps. In the second experiments, we done the profiling on just on the GPU version of the workflow to understand in which steps GPU utilized better and in which steps GPU remained idle. That in loading phase, that is the phase 1, GPU was totally idle because in that phase, it is the phase that the data loaded using CPU to CPU RAM and then go to the GPU RAM. And in some steps like PCAKN and UMAP clustering, GPU utilized 100%, and that is a good point here also because shows that because we know these steps, they have lots of matrix operations that they utilize GPU in the best way. Also, if you want to make these experiments reproducible, we should know the memory peak both sides of GPU and CPU. And we see in the GPU memory peak that's 101 gigabytes, that is 72% of the GPU VRAM. And in CPU, it's peaked like 65 gigabytes of RAM. That it shows that rapid single cell utilized GPU RAM more than the CPU RAM. Another point is that not that everyone have access to this kind of HPC level GPUs, and maybe most of us using GPUs on our, I don't know, gaming laptops that they have less amount of GPUs, and we should understand that how is this workflow is scalable. So we did a study that we started with 100,000 cells, and then by a step of 100,000 cells, we go to 1 million cells. To understand that by increase one step, how the time of the computation and how the GPU VRAM will increase. That we understood that is somehow a linear trend, that by each step it increased like 10 seconds of runtime and 10 gigabytes of VRAM. That we see that the runtime is not the bottleneck of the computation here, but GRAM at the VRAM of the GPU will be completely full at a particular step. All in all, by what we monitored, we could see the bottleneck, where GPU saturated, for example, to understand that if the runtime is a bottleneck or the memory of the GPU is bottleneck, and also what we understood, what we observed, all of on top of all the experiments that we did, is it is possible that for a practitioner to utilize GPU when wants to run the code on R and don't be engaged in lots of difficulties that may bring to re-implement everything in R, because everything needed, for example, for this particular study is implemented in a rapid single cell and just can be ported to R. The future of work, because now what we have is just a R package, and we want to do lots of tests we need to do and then make it ready for submit it to Bioconductor. Before close, I want to thank my great colleagues, Professor Sales and Professor Riso, for their mentorship, and our HPC server administrator and research technologist, Ricardo, that unfortunately this year none of them are here.
Let's give Mohammed a hand. One quick question.
I think a lot of users aren't aware of the importance of optimized blast routines for these types of things. So I was just wondering if you could comment something about the stock versus the optimized blast libraries for CPU and GPU.
Actually, it is our future work that try to understand that which version, because there are different versions of BLAST when you use CUDA or BLAST or Rapids, and we want to study almost all of them to see which of them works better.
Okay, so in the sake of time, I would ask to give Mohammed a hand again. And we will now shift to the flash talk, so I will hand over the mic. Thanks.
Then you have to reply hyvin. Miten menee? Very nice, very nice. I have had better mornings maybe. But if you go to the presentations first and then the additional information. Yes. And then there is this flash talk info somewhere. 15. Yes, that one. So if you open this. So, flash talk presenters, this is the list. And if you are presenter, please check your email and put the presentation to the drive. And I will let you 1 minute. I will have to check my notes now, so just a couple of seconds. So now, please, presenters, put your things together. So. Oh, as we know, we are the first. Thank you very much. So, in Greenland, there is the highest number of heavy metal bands in the world. So we cannot survive 4 minutes without the heavy metal. So that's why every presenter has the 4 minutes, and then it's over. You leave from the stage. And if you see, for instance, Leo with those headphones, he's not in the meeting, he's just listening to black metal. All right, so these are the rules, and 4 minutes, that includes everything. I will put some black metal, and then you cannot anymore present. Great. I guess that was the thing that I wanted, yes. So, first one is Sophie Marie, so please. On to the stage. Oh yeah, you need a mic somewhere. Oh yeah, it was there. Okay. Here. Boom, boom.
Okay, thank you. So now I really feel the pressure to stay in the 4 minutes. It's called June 5 wind over there. Yeah, so I decided because the 4CSIC talk was so good, because many people came to me and were like, oh, we used this technique in the past. I was like, okay, this is single cell DNA. I would say quite new, quite rare. Therefore, I decided to give the short talk about it. Yeah, and show a new package Scafari. Now, okay. So I will show you a bioconductor package Scafari, which we developed for the single cell DNA-seq data analysis. Oh, where's the thingy to go one slide further?
Sorry.
Yes, now, okay, now, okay, so single-cell DNA-seq data analysis gives us the option to look into the mutational landscape of tissue on single-cell level. And so the challenges are that we have a super uneven sequencing coverage, we have allelic dropout, and a lot of amplification errors because we have to amplify super, super hard. But it can give us insights into previously inaccessible heterogeneity of tissue. So for example, if you have leukemia, you can detect really rare subclones and you can look into the clonal evolution. Oh, wrong direction. It's not reacting. Yeah, really close. Perfect. So input are H5 files from Mission Bio Tapestry pipeline. We also have a Shiny app for user-friendly analysis. First step is quality control. There you can look into the sequencing coverage, into sequencing statistic, mapping statistics, and so on. Then we have the sequencing panel analysis, and it's targeted. Sequencing, you can look into the panel, the amplicon distribution, the genes, the read counts, and so on, the coverage genes, and then amplicon uniformity and performance. Then we have the classical SNV analysis on single-cell resolution. We can see which variants are present and have deeper insights into it. And then maybe the major part is that we can filter and annotate the variants. Then we can find kind of clusters or subclones and can look deeper into those because we are especially interested into some smaller clones to see, for example, clonal evolution and so on. So thank you. If you have questions, you can come to me in the coffee break. And yeah, that's it.
Very good. Yes, we don't have questions during the break, yes. And the next one is Federico, so please come to the stage.
And the thing, so I will have to resist the urge to open up the hair and enter the heavy metal. So I'm CudaMon here. So, but thank you for putting the pressure. So I have the mission to actually keep and going longer than 4 minutes for that, so that the music is triggered. I think I can talk for about 4 minutes. So anyway, I would like to introduce you briefly to a project that actually I could call an ode to PathView, that is not dead and still alive and still gives us PNG files that are pixel and then with ugly color palettes, but we still use it. Why? Because we have all seen this, and then our collaboration partners seem to still like it somehow. It's fine, but it's 2026 somehow. And 2026, 2026, 2026. You can imagine there's a picture of the PhD students in my group that is actually doing the work on this. He has been carried out the most of it. But so I don't know if you, I just, is it okay if I snap my fingers too? Just, okay. And it has been perfectly fine for a while. So, but until then, until Eduardo decided that it was time to actually have something better than that. What is better than that? Who remembers, let's say, what happens if we work with PathView? PathView probably doesn't parse all the things that you need to actually work with that. So has anyone worked with KGML files already? Has anyone have fun with working with KGML? Okay, good. So did you notice that probably the KGML files have a complete out of sync content with what the images have? So, and how do you store all the information that you require in a proper manner? So you need yes, we needed another parser. So we probably have needed a modularized parser that just does the parsing and then probably does something more than that. And instead of just downloading in every local folders and or temporary folders, we could just have used BioC file cache, which is what we did. And then instead of going from KGML into a PDF or a PNG, we can actually work with iGraph objects and move on from there. And we do this in a way that we build, render, map, and highlight things. So building, because we first need to have a simple descriptive API that can have a creation of this KEGG graph. So we first get from the KGML to the G, to the graph, and then we map the results, for example, for differential expression analysis onto that graph, so we can have continuous values onto that, for example, the logfold change. We can render that in an interactive manner, and we do this via this network. This network is a powerful, if you've never seen it, it's quite a powerful Vue.js-based implementation of this kind of library. So it kind of gives a lot of things. For example, you can actually focus onto a subset of that KEGG graph or highlight some of these nodes. Why is that? Because it's actually something that you don't just want to have like you remember the ugly picture we had at the, sorry, PathView developers, it's all good. But we can actually do better than that. And possibly we can actually return an HTML widget that has bindings for Shiny for free. So you can do a lot of these drill down operations actually in a native manner. It can just get easier to work with that. So tooltips can also deliver a lot of information. And I found out that myself, I could just be not too bad at actually extending some JavaScript code that I've never seen. But let's say I also was assisted by Claude AI for doing that. And I'm quite proud that I managed to do that. Yes, so maybe just as a tiny little, there should be a video or something like a mini demo for that. So yes, so basically, oops, or not, because it's exported. Okay, from the, it is, okay. So you can imagine that's me basically just zooming in into that, and you have probably a better color palette, and then when you hover over a node, you get a lot of information, and you can, for example, connect that node to anything that is related to that. So all the things that you can do nicely in Shiny. So I'm probably gonna put, just tried it out. My mission is over. Thank you.
Thank you. And next one is Lu Sheng. So please come to the stage. Can you find it? Great. It's this one.
Yeah, I also have this one.
Oh, does it work?
Yeah, I don't know.
Let's see. where is the mic? So?
Okay. Can I start now?
Yes.
Okay. Okay. Cool. Yeah. Hello, everyone. I'm Lu Chen from Eastern Finland University. So today we are going to talk about nanopore sequencing. Okay. So we use nanopore sequencing to study RNA modification. So what is RNA modification? So basically, there are lots of modification on RNA, and then we want to grow larger potatoes. So, so we study modification. Okay. And this is like the concept of nanopore sequencing. So you have like RNA that goes through the pore, and then you record the electric signals. Okay. And then this is how the modification looks like. So basically now, if there are modifications, then we should see like two peaks in the raw signals on the bottom that you can see. Okay, does that work? Okay, so why it's gone? Yeah, so basically there are two binds. Like one is the normal A, one is a M6A. So the electric signal at the front. Okay, so basically then the things like, okay, now how we actually see the raw signal. So this is like the basic assumption. We think like the RNA goes through the pore in a linear fashion, and then when we really check the data, it then really look like that. So basically this means like when we see the data, this will be first k-mer, second k-mer, third k-mer, and so on, right? And but then when we look at the data, it's not like that. So so we can see like there are some outliers like this one and this one. So we are questioning, okay, what are these outliers? And then we find like, hey, maybe the RNA is not going through the pore in a linear fashion, because then we will see like the outliers, they are close to the previous state, and the outliers are close to the next state. So so we can see this kind of thing all across the data, and then it means like, okay, so this is like the previous assumption, but what we think is like you should go like this way. So this is a take-home message that when you are thinking the sequencing, it's like not going through in a linear fashion. It's exactly jumping and then going forward. Okay, so these are some cartoon. This is our method called SegPol. So if you search my name, you will probably find the software. And then these are some results. So we can see like this is a previous method, and this is our method. We can really we can see clear two peaks, so it is better. And then this is another figure. So you can see on the top that we can separate the signals quite clearly, while like using previous method, we can't do that. And then this is another example, like we show like a more clear separation of the signals, while in the previous results we can't. And we can also use it to use this to estimate RNA modifications. So basically, this is a ground truth, and then this is a previous method, and then this is our method. We can actually estimate RNA modification conformation change. So yeah, so basically the take-home message is like RNA goes through the pore in a jiggling manner, and then this is like a denoising method for direct RNA sequencing. Then we can estimate the modifications. Okay, and just want to thank my student and yeah, my collaborator for this, and thanks for the opportunity.
Thank you very much. And then is Jasper Spitzer. Jasper. Mike.
Hello. Yo.
Boom, boom, boom.
Boom, boom. Okay. Okay. So, hi, I'm Jasper, and I developed a little app because I work on a lot of bulk RNA-seq datasets. And for one dataset, you often get multiple models that you want to compare and contrast. And I wanted to be able to do that and share it with collaborators or other people and discuss and communicate these results. The general idea is you just process and analyze your data like you would usually do with all the tools that you like, and then you use a structure that already exists in the DD experiment. If you like that, to store those results, then you load just that object into the Explorer, and then you can have a nice look at it. So that's the general interface. Um, by default, you get a view of like metadata. So right now nothing's loaded, so there's demo. But then you load in the object that you care about. Standard, you look at call data and other metadata that you can pick what. Oh, sorry, yeah. You select the comparison that you want to be looking at, and then you have different options. You can look at gene expression, but also different plots, the DE results themselves. So you get, like, for example, a volcano plot, and you get the enrichments that are stored in the objects, and you can switch between the different models and comparisons. And yeah, that's it. It was originally just a tool for me, but I figured some people might get a use out of it. And it still needs some improvements, especially visually. Currently, it's not on Bioconductor. I don't know if it's in scope, but it's on GitHub, and you're very welcome to use it. If you have any questions, come up to me, ask. That's it.
Thank you. Very nice, thank you. And then is Giulio Benedetti. Where is Giulio? I can hear is this.
We'll just click on that. Thanks. Yeah, no problem. Submission.
Yeah, it's coming there.
Have to listen to heavy metal again. Thanks. Oh, it doesn't let me switch, but okay. So if anyone wants to go beyond just colorblind friendly color scheme, this presentation is for you. If it lets me switch. Oh. Here it is. All right. So after this presentation, you have probably more tools to work with um, accessibility for your research output, posters, and things like that. Last year in the European Union, there was a new law passed that products have to follow this kind of guidelines for accessibility, and now companies have to really follow them if they want to be competitive. So perceivable, operable, understandable, and robust products, software we're talking about here. And for the accessibility wise, this means that when products should follow should have a way to be accessible to not just individuals who can maybe see, or maybe also those who can just touch or just hear information. So they don't have access to all senses. And one possible solution is to go for this multisensory visualization. And that practically means to use multiple senses or sensory modalities to encode the information in our graphics, for example. And this isn't useful only for people with disabilities. It's actually demonstrated that encoding information across multiple modalities helps also remembering that information or learning it. So these are a few examples. Probably we won't have time to go through all of them, but not again. Alternative text may be more familiar to most of us, and there is a package in R to produce R alt text for our graphs. Someone developed an algorithm to sonify, so to represent hierarchical trees or phylogenies with sound. So you can look it up if you're interested. But not just plots, all graphs can be sonified, so using sound to encode them. Another option is this tactile paper, which is a bit expensive and requires a specific machine to produce, but then people who can't see can actually feel with touch what we are talking about. And this is a book written by NASA to describe eclipses by touch. 3D printers. Then there is this, in the past, it was conceived as a form of art, lithographs, but this paper shows how it can be used to represent also quantitative data, such as chromatography assays. And then finally haptics, which is using this keyboard that people can feel through this, I think, vibration. It feels like touching objects with some kind of like 3D surface. And it's available in iPads, for example. And even your phone might have this mode if you look through the settings. These are a few useful plot packages in R that allow you to do this. Plate by R was really cool. It's deprecated. Someone wants to pick it up, would be really great. Doing ggplot stuff, but sonification. Yes. Yes.
Thank you. And the next speaker is Damien Kaukonen. So please.
I pray. The one my friend uploaded to it. Yeah, that one. No, yeah, that one. Hi, my name is Damien. I decided to be mad and present advanced statistical modeling and breast cancer to a diverse audience in 4 minutes. So hold on to your butts. Just a bit of overview. Going to do a bit of study design for background, but I want to focus today on what we saw when we did 2 different survival modeling. So we looked at recurrence-free survival in breast cancer. The take-home slide. This is a big slide, but the take-home message is we started with a large study population, which was every woman diagnosed with breast cancer in the Stockholm-Gotland region from 2007 to 2020. We had a few exclusion criteria, but we still maintained enough people to do statistical analyses. With our study designs for exposure in breast cancer, you have 2 kind of prognostic markers. There's several, but the 2 main ones are human epidermal growth factor receptor 2 and estrogen receptor. So if these are either amplified or overexpressed, we say they're HER2 positive or ER positive. Otherwise, they are negative. Our outcome was distant recurrence. That means the breast cancer came back at a distant location, not just within the breast, or death from breast cancer, because if they died from breast cancer, the cancer must have came back. And their follow-up time is from diagnosis to either event, they were censored, or the end of our study is June 15th, 2020. So at first glance, we run a Kaplan-Meier, and what we can see that HER2 plus does worse than HER2 minus in all patients and among ER positive patients. And of course, you can easily just say, this is our publication, call it a day, right? We got statistical significance, they separate. But we didn't answer our question of how ER and HER2 interact. So at first we thought we would do a Cox, but as anyone can tell, these are generally not linear plots. So we actually introduced this is why I want to introduce one of the concepts that we use, called splines, which we use in flexible parametric modeling. So what a spline is in maritime languages, when you want to bend wood, you add weights to it. to bend the wood in a certain curve. So now we do that to plots to bend our linear model to make it more parabolic. This allows for inflections, and these inflections are determined by degrees of freedom. And here we use 3 degrees of freedom, so we allowed 3 bends in our plot. But another thing that we noticed when we were looking at our population, and it's nice that it's cut off, oh no, it's not cut off, is that HER2 positive seems to be significantly associated with higher tumor grade, larger tumor size, and being lymph node positive. So it means they're more likely to spread to lymph node at diagnosis. Therefore, they're associated with worse tumor characteristics, which are also associated with worse survival. For a point later, HER2 is also associated with interval cancer, which means it occurred between 2 screening rounds, which means it's really aggressive, that it wasn't detected in screening, but the woman herself had symptoms, went to the doctor, and got diagnosed. That's a point for later, but we decided to adjust for these tumor characteristics and treatment. And what you see when we do our adjusted survival curves, they now switch. HER2 plus is actually better, has better survival than HER2 minus. And what this kind of tells us is that HER2 is just acting as a proxy for these worse tumor characteristics. And we also notice that there's obviously a time varying effect, and this time varying effect was actually modulated by ER status. So the kind of take home message that I wanted to take from the study was, if we go with the Kaplan-Meier, we see that HER2 plus is worse in all NER patients than HER2 minus. That would tell us that we should focus on treating HER2 plus more than HER2 minus, that it's not that good. But if we do the adjusted flexible parametric modeling, we see that HER2 plus actually has better survival. And our conclusion kind of there is that, as I said, HER2 kind of acts as a proxy for worse tumor characteristics. And therefore, we would then need to focus better on screening for these patients. And this is backed up by the fact that they are more likely to be interval cancer. So it's kind of interesting that your model selection heavily determines where you're going to put your future resources and your future research. And I say spoiler alert, because obviously now my research is now focusing on understanding what happens in these HER2 positive patients prior to diagnosis using mammograms and all sorts of other data. So this was surprising to me, coming from biology, going into epidemiology and coming back, is that you really need to explore the data in order to select the proper model. And that's everything. This is the paper if anybody wants to see it.
Thank you very much. And the next one is Mehrad Mahmoudian.
Great. Here, thank you.
There.
Yep, magnificent. It's the last. Yeah. You don't need to download, just double click. Yeah. Yeah, so we basically pulled forces. So Arthur and I, we're touching on the same topic, a bit different though. So please, before the presentation begins, it's a bit QR code heavy because of the time, so have your phones and cameras ready if you want to. So let's give it a try. So basically, we're going to talk about reproducible analysis, R, Python, Perl, whatever environment, because at the moment, we have a bit of a situation. So basically, why to bother? Because basically, you install the software, then you do your analysis, and it takes time. It takes months or years. And then some stuff like CRAN or rolling release, and something like Bioconductor is point release. And if you use other package manager, then that also has its own kind of way of releasing the software. And then there is also the package dependency that can be very deep, and also multi-languages and multi-platforms. And then, yeah, well, the analysis can take ages. So your computer gets updated over and over and over. So basically, every time you're analyzing your data with a bit of a different software, different R, different packages. And the problem is you cannot publish your laptop. It's not a paper. So, and also the last bit is that if the researcher is lost, for example, if I die, my colleagues can easily boot up everything I have done, the exact environment, and then go forward with it. So basically, there are already some solutions, quote unquote. There are these classic package managers like the PackPack R package, the Cargo for Rust, Go, pip for Python, and etc. Not good. There are a lot of literature to read about. You can also do the Git repository as submodules, which is a little bit better, still suboptimal. You can also have these local environments like the VN, RN, and the old school packrat, still not very good for obvious reasons. There are attempts like the rig, which is written in Rust. It's an R installation manager, and also it can install the R packages and also boot up your R Studio with a specific R version. Still doesn't address the question of, for example, Fortran and stuff that you install while updating the softwares. Then there's the container like AppTainer, Singularity, Podman, Docker, and also possibly AppImage, not optimal. There is a very nice YouTube video about a NixOS presentation that I can share the link later if you want during the coffee time, why containers are not very good. But there are attempts like Rocker and R2U by Dirk that they're actually doing a very, very nice job. Still, it suffers from the container problem, but it's basically second to the best in my opinion. And then we can go to the declarative environment, which we can either use something like Nix with Rix R package, or we can go with Geeks. Arthur is going to cover this, the first bit, and then the Nix is basically what I'm going to quickly cover. And also, by the way, this QR code up there, it's going to stay until my end of my presentation. That is to my blog post about how to set up with all the codes and everything. So just copy, paste, and enjoy. And sorry for that bit. Yeah, so that QR code is not readable, probably. Thank you. So, yeah, so the solution is basically Time Machine. Majority of people probably think Time Machine is this one. Some nerdy people might think it's that one or that one, which that is the nerdiness scale. But that is the nerdiness I'm talking about. If you know what that means, then you're my type of people. So basically, my best solution is basically using Geeks to provide the Time Machine. Basically, you're not going to deal with it directly, but it's going to be there for you. And then there are a bunch of bash, very simple, very short, less than 50 lines. Actually, it can be single line. And that can automate things and handle the environment creation and yeah, to make things orchestrated. This doesn't go. This is yours.
Oh my God. All right, I have no idea. I just looked at these slides just before. Yeah. I also don't know what to cover here, so I will just skip it. Yeah. So basically, I will be presenting Nix. He talked about Geeks. Nix, basically same idea, but it does not follow the file hierarchy as in Linux. And Nix is actually a language, and it's a package manager, and it's also a Linux distribution. So the idea is that every single package is completely isolated from the rest of them. So you never encounter a dependency hell because every single package is built independently, and it depends on its own dependencies, and then dependencies depend on their own dependencies. There are problems, cryptic errors. Sometimes with Nix, it's really difficult. You don't really understand what's going on, and you also have to learn the actual language. But there is a solution to this. If I go, so most of you already know R, and then you can basically combine R and Nix and write R code to generate completely reproducible R and Python development environments with an R script, which then generates Nix code. With this package called Rix, which was developed by two gentlemen, Bruno Rodriguez and Philip Baumann. So the idea here that we use this function and we declare the R version, for example, the R packages, and you can also declare the versions of these packages that you want. And then we can go a step further. So we can fix the development environment to a certain date, and this environment will be guaranteed to be reproducible if I share this code with you. It not only allows to use R packages, it's also for Python. So you can declare the Python version. We can go next. We can go even further, so we can also declare system packages. In this case, it's Quarto and Git. You can add pretty much any software that is available on a Linux operating system. You can even declare the IDE. So in this case, it's none, but you can, for example, if you use RStudio, then you can say, I also want to use RStudio, and then you can use RStudio with exact same version that I'm using it. Yes. Come on. Okay. And you can go even step further, and you can actually download some packages directly from Git with a specific commit, and it will be guaranteed that this specific commit will be used when I share this environment with you. And then it will generate a very strange default.nix file, which you can look at, and it's like very cryptic language. But I think most of you will figure out what's going on there. So yeah, they wrote a paper, and the package is available on CRAN. So you can scan the QR code later. And yeah, thank you very much. You can connect with us, and we can discuss these development environments with you.
Thank you, Artur. Thank you, Mehrad. And the show will be finalized by Lieven. Please.
Thank you. I will just use the system of FEDE, which works much better. And I will also build up on the poster of FEDE. So it's all about FEDE today, but I will invade. So who likes single cell experiments? Who likes the alt experiments and single cell experiments? Who shares my pain that we cannot visualize, that we do not see these alt experiments with IC? So I have a dream for mass spectrometry-based proteomics. And in mass spectrometry-based proteomics, we typically try to prioritize differentially abundant proteins, but I do not see these proteins. I see precursors or I see PSMs. And luckily we have this nice Q features architecture. We also had a poster of it from somebody, from Leopold, from the group of Laurent. And we can read in our PSMs, our precursors, and then we can start to normalize them and to log transform them and to summarize them. And each time that we do a step with that, we get a new set. And all these sets are stored in the same container. And finally, we get our protein abundance values. And all these sets are linked to each other. So if I take a protein, I can basically get back and I can get all the raw data of it. And it would be great if we could visualize it. And if we add MSQROP2 to that, our package, we can also do differential abundance analysis. So I would like to explore that with IC, and that should be possible. I could take my proteins as the main experiment of a single cell experiment, and I could put my peptides and my PSMs and so on in the alt experiments, and I could link the two experiments to each other via the protein groups file, a protein groups variable that is used to summarize the PSMs into the proteins. And then I could select the protein in IC that is differentially abundant, and if I could see these alt experiments, I could also see the peptides and the PSMs. So this is not possible because I do not see alt-exps. So therefore, I decided to crash the IC party. And it's totally in line with the theme of the talk. So it's something that we basically came up with in a short course on proteomics data analysis. And we brainstormed with Charlotte, Robert, Florian, and Atime in Basel. And then we suddenly talked about having a kind of a new layer to visualize this alt-C experiments. And we also dragged the Fede and Kevin into the party. So what have we done? We basically Charlotte developed a kind of a first version of the alt experiment layer as a kind of a new panel, and we are now at version 6. And what can version 6 do for you? This is what we get. We get a kind of a volcano plot. We can select our differentially abundant proteins. We get this nice table. I can click one of these proteins. I can then see like the abundances of the proteins. And because we link these proteins with the alt experiment plot via the protein groups, I can also see all the underlying precursors that are there. So that is, according to me as a proteomics researcher, very cool. What can we do more? We can also load multiple assays in this alt experiment. So I can also go from the normalized precursors to the log transformed precursors. So I can basically visualize the entire data and I can see how each of the steps that are nicely done in Q features are actually behaving. So do you also have dreams for alt experiments? So maybe you have other suggestions. So we will develop a new package for that soon. I would have loved to have the music because it fits in the theme.
Thank you very much. That was awesome. And then if you can, Jeba, put additional information. First, clap to all the presenters. And then do this. So now we have break. And after that break at 11 or something, starts these birds of weather sessions. They are very informal discussion thing is, so you can take coffee there also. So it should be fine, and they last 1.5 hours. And yeah, these are the rooms. So there will be Finnish bioconductor community discussion in ministry. Here will be how can the bioconductor community help early career researchers, and in Tellervo there will be embedding environmental sustainability in your bioconductor package. So thank you, and this was my friends.